This part of the course is going to have a look at monoclonal antibodies. So you're going to have a look at what monoclonal antibodies are, how they're used in particular in treating and diagnosing cancer, pregnancy, and blood clots. So the first thing to remember then is that an antibody is something that's produced by a B lymphocyte. Now, antibodies can obviously be really useful, and it would be really great if we could take those lymphocytes and reproduce them. Unfortunately, it's not that easy to do that, so something else has to be done. So the first thing they do then is they need to produce the B lymphocytes, and to do that they will inject a mouse with some of the antigens. Now, antigens are the things that the antibodies latch onto that are attached to the pathogens. So they will inject these into the mouse, and the mouse will then produce antibodies. The second thing they'll do is take some tumour cells. Now, tumour cells divide really fast, but they don't contain antibodies. So what they'll do is they'll start to grow those in a lab in a petri dish. You will then have B lymphocytes from the mice, and you'll have the tumour cell, which quite easily reproduce. And what they'll do is they'll fuse them together. So they'll take the lymphocytes, they'll take the tumour cell, fuse them together, and they'll produce something. Now, that something is called a hybridoma, which is basically just a hybrid of between the two. This hybridoma then will be put back into a petri dish and will be allowed to divide, and they divide really quickly. And what they'll do is they'll produce monoclonal antibodies. So monoclonal, mono means one, so one antibody, and clonal means they're reproduced. So a monoclonal antibody. So now we're on to the uses of monoclonal antibodies. Now, the key thing here is that these antibodies can be made to latch onto anything, to bind to anything, which makes it really useful in, for example, pregnancy tests. So when pregnant, a hormone is produced by women that is only there when they are pregnant, and pregnancy tests can detect this hormone. So a pregnancy test will have two parts to it. On the right it has a test strip which has antibodies stuck down. On the left it has antibodies which are attached to blue beads. So when you pee on it then, if you're pregnant, the hormones that are produced when you're pregnant get stuck to these antibodies. Because the antibodies are stuck to the blue beads, they get carried down the strip to the test area. This test strip will then turn blue, which shows a positive result and shows that you're pregnant. If you're not pregnant, however, you aren't producing those hormones, so there's nothing to get stuck to the antibodies and the blue beads. It doesn't get carried down and it doesn't turn blue. So the next use of these antibodies then is the diagnosis and treatment of cancer. So cancer cells themselves have antigens on them. Now, these antigens can be picked up by antibodies. However, the body doesn't normally have a way of treating them. So what they'll do is they'll use monoclonal antibodies. They'll prepare things that can be used to find and kill these cancers. So the first thing we're going to have a look at is diagnosis. What they'll do is they'll take a monoclonal antibody and they'll give it a radioactive element. Now, what happens is it's given through a drip, put into the blood, and it goes through the body. It'll then attach onto these cancer cells, and then when you do take pictures of it, then it will show a bright spot on these pictures. This will tell you where it is, how big it is it spreading, and so on. And then they can go on to treatment. So what they'll do is they'll put anti-cancer drugs onto the actual antibodies. Again, it's given through the drip, and it targets just those specific cells, which is great because it'll only kill the cancer cells and it'll leave the normal cells alone, which obviously means it's got less side effects than using radiotherapy, which can kill all cells. So the final thing we're going to have a look at then is blood clots. If you'll excuse my appalling drawing of a foot, and yes, it is a foot. When blood clots, the proteins in the blood join together. Now this forms a mesh type thing which stops any blood and anything from getting through, so it clots the body. So what they'll do is they'll produce these monoclonal antibodies again, and they'll have specific things on them and with radioactive elements, and that'll tell you where the clot is. It will attach onto the proteins in the clot, and then when you do take that picture of it, it will give you that bright spot, so then you'll know exactly where the clot is.